Structural biology has enabled the design of new treatments. So in the case of HIV, it's really the knowledge gained from structures, the components of the virus that has enabled the invention of new drugs. This has been a decades-long effort. Of course, similar efforts are now underway for SARS-CoV-2. And this is just to introduce structural biology as an important technology and thing that we work on as researchers, as a research community. In recent years, cryo-EM has really accelerated this process. So the amount of time that it took to get the structures of the SARS-CoV-2 proteins and viral components was much faster. And so cryo has advanced to the point where we can really get atomic structures of many proteins quite rapidly. And it's fantastic for large, rigid targets. So my lab's interested in extending the benefits of this amazing method to targets that are kind of below the resolution limit of the technology. And it turns out that there are a lot of targets that fall in that range. So we're interested in small, flexible targets, proteins, and nucleic acid complexes. So what do I mean by small? Well, about half our proteome is, quote, small proteins. So basically a little bit like less than 50 kilodaltons. That's about 4,000 atoms. And if you look at the genomic distribution of protein size, we see about half of our genome encodes proteins that are smaller than this. This doesn't take into account oligomers. Of course, those are very common, but just from the size of the ORFs. So, and I'm not going to talk about flexibility just yet, but just these small proteins are sort of essentially invisible to cryo-EM without some special trick. And we and others are working on figuring out tricks at using nanotechnology in order to make these proteins visible. So, so here's an outline of my talk. I'm going to describe the cryo-EM method in general, just to introduce it for those who are not familiar, and then why visualizing small proteins is difficult. I'm going to talk about the approach that our lab recently published, and then I'm going to talk about the future vision or the next phase of this project, which is we're able to see small things. Now we need to constrain flexibility. So I'll talk about that at the end. And that last part is part of a collaboration with Bret Victor. So I'm going to hand it over to Bret, and he's going to describe our early progress on that problem. So starting with how do we visualize proteins with cryo-EM? So here's a quick crash course in cryo-EM. So imagine that you want to solve the structure of this protein here. Pretend that you don't know it's a duck. We know it's a protein. We want to find the structure. So what you would do is spend a few years doing biochemistry, preparing a pure sample. You load this sample onto an electron microscope grid. It's a little metal disc with some holes in it. And if we zoom in here, what we want is a monolayer of particles. So we're going to add our droplet, blot away the excess liquid, and then quickly freeze the sample. And so this is done with an instrument called a Vitrobot, which resembles this monkey with cymbal toy. And that's actually really what it looks like. So here's a movie of this blotting and then plunging step. And so now what we have, so we've plunged the sample into liquid ethane, so it's frozen in vitreous ice, and we load that specimen onto our electron microscope, and then we shine an electron beam through the specimen onto a camera that can detect electrons. And so we get the shadows of all of those individual particles on the detector. And so we collect thousands of micrographs over the course of a day or two, and then we can pick particles from that data set, so identify those shadows and then kind of crop them into individual images and then align them together into classes. So each individual particle is quite noisy and low contrast, but if you add them all up and make a stack, then the noise kind of averages out, and where there's actual density, you can see the particle. So the last step is to take these different shadows, the classes of shadows, and then align them in 3D and do the 3D reconstruction. So that's basically, in a nutshell, what cryo-EM looks like for a nice big rubber ducky. But what happens if you have a smaller duck? So why are smaller proteins hard to resolve with cryo-EM? So this is due to particle misalignment at this reconstruction step. And so what I mean by misalignment is that we have to kind of guess and check what the 3D angles are for each of those shadows. And if we guess wrong, then we don't get an accurate structure. And so just to dig into how this works a little bit more, to build this 3D structure, we basically take all these different 2D particle images and then build a 3D model using this iterative expectation maximization procedure. And so this is expected to converge over time. So basically you sort of make a model, you kind of guess, and then you check, does this look good? And then you jiggle the angles around and check it again. And if it gets better, then you continue in that direction and eventually converge on something. So when the particles lack the asymmetric features that you need in order to do that alignment, then the algorithm stalls, right? You converge on something, but you don't yet have a high resolution structure. So a little blob that's like, the sub pixel size, if you flip it over, it still looks like a blob. You're not going to be really able to align that. So that's a challenge. And so we asked, well, what if we could determine the true orientations of the particles before we started this model building? They're too small to really work with at that stage. And so we thought, well, maybe this is a job for nanotechnology. Can we build a nanomachine that resembles or has the same purpose as a macroscale goniometer, which is a device that can precisely, has a stage and you can kind of set one or two, like an angle, the precise angle of the stage or the orientation on multiple axes. And so what we want these goniometers, but for molecules. So that's what we've been working on for several years now. And I want to just say that this work was primarily led by a former postdoc in my lab, Terrell Axel. Then we collaborated with our neighbors at UCSF, Zanlin Yu, who's in Yifan Cheng's group. And so this has been an iterative process to build a working version of this instrument, and every 2 years we got a little bit better. So starting with no protein on the device all the way up to a pretty nice structure a couple of years ago. We've been using this protein called BurH, which is a tail-like DNA binding protein that's relatively small, and it has this helical pseudosymmetry, so it's actually a very hard target for cryo-EM because if you flip it around, it looks identical and sort of at low resolution in both orientations. But it's a very well-behaved protein. So the way that our device works is that we load a big excess of those proteins onto our goniometers in solution, and then we put them down on the EM grid. And so we use a graphene oxide support layer, so they all lie flat. And then we kind of freeze them and go through the normal cryo-EM process. And so what we have is a bunch of particles where each has a single protein bound to the stage. And the way we control the angle is that if you think of this stage as like a little clothesline or something, we can like feed through the DNA and shift the register one base pair at a time. And as we shift the register, the binding site kind of changes its orientation relative to the chassis. And so as we step through, we get a series of different angles. And so we use a barcode. In this case, this is from the previous generation design, but we have a large asymmetric feature so we can identify each of those particles because the protein is still too small to see even when it's on the stage. So we can collect our data set and then sort all these into class averages, and then we can start cropping out the origami and zooming in on the protein. And so you can see that this sorts as expected. And then we can use these large features to identify whether the goniometer is face up or face down on the grid. And yeah, so just to revisit the reconstruction pipeline using this device, what we end up with is we built 7 variants of the goniometer, each with a different angle. We feed those through this algorithm, and then we collect these 2D classes that are not just the 2D class, we also have the angle information from the goniometer associated with it. And so we feed that part into the algorithm. And we had to tweak the software a little bit to to make it recognize and use that information. But at the end, we end up with a nice 3D map. And just to show you that this actually works, let's compare this structures that we get if we don't use that angle information. So what we're showing here in the cartoon ribbon diagram is the known structure from a crystal structure of Burr H, and then the density is basically the predicted structure from the cryo-EM workflow. So this is using different software tools. It's basically a coin flip of whether they get the orientation correct. The tool Relion gets about 80% of the orientations correct, but it still has some issues with the density. And then this is Relion plus the origami. And that ended up being the highest resolution map. So that was nice after several years to finally find out if that worked, which it did. So now we're moving to the stage where we want to apply this technology to study proteins of unknown structure. And we're not quite there yet. I mean, there may be a few easy structures that we can look at, but most of the structures are going to have some flexibility in them. And so we're really interested with our collaborators to study nuclear receptors. And so these are the molecules inside your cells that kind of respond to signals and then turn on gene expression and so on. And so there's dozens of different nuclear receptors. Nobody's ever succeeded in solving the full-length structure of a nuclear receptor using any method. And so here's an example of, from where you actually can see there's like these dotted lines here. So people can like chop these up and solve parts of them and get an initial model. But this is kind of the type of structure that we want to look at. We could generalize this hopefully to non-DNA binding proteins in the future, but this is where we're starting. And so what we're doing, well, before we get into that, we need to validate our new approach and build a new set of goniometers to actually do that, not just bind the protein, but constrain it. And so we are, again, using a protein of known structure, or we actually want to design a fusion protein where we know the structure, so we can use that as a test case. So we're going to, again, start with Burr H, and then right now we're thinking of fusing GFP, which is a nice small little domain. We could add a flexible linker and connect it to Burr H, and that could be like our test protein or a control that we would use to engineer a goniometer that could both bind the Burr H and then somehow lock that GFP into a stable conformation so we can see it in the final reconstruction. So I'm about to hand this over to Bret, who's going to talk about actually what we've been doing with that part of the project. But I just want to acknowledge that this is a software and simulation themed conference or workshop, and I haven't really said much about software. Needless to say, software is deeply involved at every single step from the design all the way through the data analysis. And it's also involved in a lot of the steps that we don't talk about. So I just grabbed a few screenshots of different tools that we use. So we use software to design our molecules, to analyze our cryo-EM data, but we also use it to manage our lab notebooks or present to each other. So we have slide presentations, or when we write our papers and reference other papers, there's specialized tools for all those things. So we're currently in the process of reimagining what software and computation could look like for all software that we touch in the lab. And so I'm really excited about this. Hopefully you'll share that excitement out in 10 minutes. So I'm going to hand it over to Bret so he'll talk about what we've been working on.
Thanks, Shawn. I'm going to be showing a prototype of a biomolecular design environment that we've been working on for a couple months now. And it's based on a new kind of operating system that my lab's been developing called RealTalk. And RealTalk runs in the real world instead of on screens, and you'll see what that means in a bit. And what I'm showing, these are just prototypes, right? Their purpose is to explore ideas for what Shawn's lab could look like in a couple of years. They're not really working tools yet. That said, a lot of this actually does work. We're hand-waving some of the simulation and analysis parts, and our protein engineering is pretty simplistic, but the stuff that looks like magic, that's all real. And it's pretty hard to follow from a talk, so we're inviting you to come by the lab at UCSF and check it out, talk to Shawn. We'd love for you all to come by and get your hands on it. I'm going to be showing Shawn and me going through some tasks from the project that he just described. We're going to be designing the fusion of two proteins, BRH and GFP, designing a DNA origami structure to hold the proteins, and making the origami in the wet lab. And this is the space in which we are working. So first, we want to get the BRH and GFP proteins onto the table so we can start playing with them. So here, Shawn is typing BRH into the PDB search card, and these are the two search results from the protein data bank. I'm going to pick up the one we want using one of these mock test tubes. It's kind of like a game piece in a board game. And so now the 3D structure's on the table, and I'm just going to use this laser pointer to spin the molecule around, examine it, check it out. And now I'm scanning through the sequence just to see where those residues are in the structure. And I'm going to look up the GFP the same way. And I'm going to grab the one that we want. And now we can just play with them, kind of positioning them relative to each other, thinking about how we want to connect them. And in order to connect them, we'll need to permute the GFP, which means closing up its termini with a linker and breaking the loop somewhere else. And so this is a little permute program that we made, and I'm pointing its whisker to where we want the termini to be. And we're going to pick up the permuted GFP in another tube. So that's our new protein. I'm just comparing it to the old one, and I'm going to swap it into the scene. Now I'm going to bring out our splice program, and I'm going to point its whisker to a spot on BRH where we want to splice in the GFP. And I'm going to pick up the protein representing the two of them spliced together. And here I'm scanning through the new sequence. The blue text came from the BRH, the green text came from the GFP, and pink is the linkers that we added. Here, Shawn is showing me a more complicated idea that uses two BRHs and a spycatcher spy tag system to pin down the GFP in two places. And he's kind of just doodling on the table here. So that's a future project, but even for now, with one BRH, we don't know which splices will actually work. So here we're trying a different splice point, and we're staving that off into its own tube. So we have these candidate designs on the table, and one way we like to plan an experiment is by envisioning the gel that we expect to see at the end. So here we're organizing our mock tubes on a mock rack, and we see them as lanes on a simulated gel. And this includes both the new proteins that we want to make as well as intermediate products that we'll want as controls. But before we think about protein expression, we want to design the DNA origami structure that these proteins will bind to. So here I'm swapping out the scale bar on the table for one that's more origami scale. The protein's pretty small at the scale, and we can start to imagine the goniometer around it. So we bring out our tub of origami blocks. Each of these blocks represents a pleated layer of DNA helices, and the blocks snap together with magnets at valid crossover positions to form a honeycomb lattice. The blocks I'm showing here are 6 helices tall and 6 DNA turns long. Here we're going to be assembling the blocks on the table into a 3-dimensional scale model of the nanostructure. As we build the model, we see the routing diagrams above fill in with an auto-routed scaffold strand. Here we're putting the blocks for the single helix stage that the proteins will bind to. So this is one goniometer design, and Shawn's going to pick it up, and again, into a mock tube. And I'm playing with positioning one of the proteins that we made onto the stage. Here, Shawn is adding a feature to the bottom of the origami in order to make a variant. He's going to pick up this variant into another tube. And now we've got just a few designs on the table, and we can spin them around, we can check them out, we can discuss them, we can point to things and talk about them. Here we're organizing the ones that we want to make on a rack, and again, seeing the simulated gel. But in this case, we also want to preview the protocol that we'll be using to fold them in the wet lab. So I'm putting out a couple more mock racks and mock plates. And the staple strands in our set of designs are automatically assigned to wells in the plates on the left. And we see an auto-designed folding protocol flowing through the tubes to the right. This is exactly the same setup we'll be using for real when we get to the wet lab later, but now we're just simulating it. Here I'm going through the protocol step by step to preview each pipette transfer. And we see at each step the contents of each tube and their concentrations. And this is all live, right? As we rearrange the rack, we get a new protocol. And here we're rearranging the origami blocks and seeing the staples and protocol update immediately. Now, our purpose in designing these origami structures is to be able to recognize them under the microscope. So here I'm putting it on the mock EM grid. I'm putting one of our mock tubes on it and firing our mock electron beam at the grid. And this generates a set of simulated micrographs. So up on the wall, I'm pinning the Show Micrographs program, and we're looking through our simulated images. As we put more samples on the grid, we see more species in the images. Here we're adjusting the concentration of one of the samples to change the density. We can use these simulated images to design our image analysis pipeline. Here I'm putting up programs that would pick particles from the images, classify the particles, show class averages, and so on. Here Shawn is rearranging the origami blocks, and we're seeing that that entire analysis chain updates live. The idea is that we can design our analysis algorithms at the same time and in the same place that we're designing the nanostructures to be analyzed. And because it's simulated data, we already know the right answer, so we can evaluate how well our algorithms are doing and get everything debugged in simulation before going through real data collection runs. So we decide on our origami designs, we order algos, and we're ready to fold them in the wet lab. Here we're putting down our generated protocol on the bench, and it's prompting for the plates and racks that we'll need. We get those, we get out our tubes, get our reagents. Here I'm lasering the names of some of the reagents in order to see information about them, including where they're physically located in the lab and where we can order more if we're running low. We're going to do a quick preview of the protocol to remind ourselves of the steps. And then we begin. At each step, a projected line shows exactly what to pipette where and how much. Next to every tube, we see the expected contents of the tube and their concentrations at that point in time. Above the bench, we see the protocol as a list of instructions, and we see where we are in the list. So this is kind of like turn-by-turn navigation for the wet lab. Now, at the end of the project, we might put some of these tubes into the freezer, and maybe years later, we take one out, put it on the bench, and we would expect to see immediately from that physical tube the protocol that generated it. We could also see maybe the origami designs, the data we later collected, the paper that was published, all that could be attached to the physical test tube. So physicality is one of the key concepts that we're leveraging here. Using our hands, designing 3D structures using 3D models, seeing what's physically in test tubes. Molecules in test tubes are physical things, so we'd like our computation out in the physical world where the science is happening. Another key concept is the social nature of this environment. People work together, side by side, getting their hands on the same things. What if this is what a group meeting was like? People getting together to play with each other's molecules. A third key concept is designing using computational models. If we can simulate molecules, simulate nanostructures, simulate microscopes in real time, we get immediate feedback as we design, and we have a consistent shared mental model with others. And a computational model also serves as a precise representation of what we think we understand. If we do a physical experiment and it doesn't match our simulation, then we've learned something. We can update our models. The last key concept is that this is a completely programmable environment where programs can be quickly improvised in the moment. So here we're using a different linker in our protein splice by directly editing the splice program. Here I'm making a new scale bar by editing an existing one. And here, we're directly programming an origami block to stimulate passivation. This entire set of tools that you've seen was made in exactly this way, by editing the pages on those posters there, any of which can be changed live at any time by anyone right while we're using them. What you see on these posters is the entirety of this prototype. There is no code base in GitHub. Everything having to do with biomolecules is literally just these 20 or so sheets of paper. Anybody can grab them, anybody can edit them. Of course, we're also using the RealTalk operating system. That's its own poster gallery, which is a bit larger. Although lately we've been keeping our own copies and binders. So the whole operating system itself is made of a manageable set of physical objects. It's completely live editable, completely browsable by anyone. My lab has been working on RailsTalk for several years. These are the core people involved in that work. This biomolecule prototype was made by the three of us just in the last couple months. It's been a lot of fun, and we're looking forward to seeing where it goes. Thank you.
I've seen in a long time. That was truly spectacular. But you've left out the materials properties. I mean, how is the page reprogrammable? How is the page read? I mean, what are you using to make all that happen?
Yeah, that's on that poster gallery that I showed at the end, or like I said, now we're keeping that in binders. So the operating system itself is made in itself out of physical objects that are recognized by the operating system itself. They're recognized by the visual properties. We don't really have time now to go into how the system works.
What's the camera projector that you're using? Like, you've got that online?
Off the shelf.
Off the shelf,
Yeah.
Okay.
Have you guys considered including sources of experimental error when you're generating those protocols to see what uncertainties you might expect as they go through the protocol?
We'd love to do that. That was actually a key feature of one of Shawn's earlier works, which was a gel simulator, gel box. And like one of the interesting aspects of that was it simulates the things that can go wrong when you're running a gel.
We want to do everything and see it, basically. I mean, that's all of it.
Thank you. Very nice work. It was very impressive. One question. It seems that then a natural extension would be just to add robotics to do the performance of the
Absolutely.
actual work, right? So I wonder if you're thinking on that direction. And then the second question, I don't mean it in a negative way, but I feel like I have to be that guy to ask the question, does this crash and how often does it crash?
So the first question, in our lab, yeah, in the past we've done a lot of, some experiments with robotics. We have a robotic table where you can wish for objects to move around. The idea of the operating system is that it'll use whatever sensors and actuators you have to gather information about the world and make the world the way you want to. So what you saw here was a lot of projection, but it's kind of built into the operating system that it's kind of about like looking at the state of the world and then changing the state of the physical world. The question about crashing, when a virtual world crashes, you lose everything. It's all gone. When this crashes, then the paper's still there. You can still read it. It actually just comes back up in a couple seconds. This is kind of a whole other talk, but when you're working with 90% physical materials and just 10% computation, there's a lot less can go wrong, and when things go wrong, they're much easier to fix, because the complexity of the system is way simpler than any operating system that we currently use.